Great. It's really good to see all of you. I really want to thank you for your participation today and also for those on Zoom. My name is John Sferrin. I'm at the University of Wisconsin where I have a lab that studies peripheral nerve and CMT. I'm also very privileged to serve as chair of the Scientific Advisory Board for the CMTA. And I get to work with a lot of great scientists, many of which you will see today. And I also serve as a member of the board as well. So our purpose today is really to cover CMT1. The major types of CMT1 are CMT1A, 1B, and 1X. And I want to give you just a quick overview of really how we really have a multidimensional approach to trying to bring new therapies to this disease. And this is a diagram where what you'll see today during this session is a series of initiatives, some of which are genetic therapies, ASO and gene therapy. We also have candidate drugs that are being tested in preclinical testing as well. And some of these ideas come through our network of scientists and clinicians. But we also are very keen to work with companies, and company partnerships, which you'll also see in our session today, are really a key part of how we want to really attack CMT from various sides. And then the last thing that I'll talk about, and also subject of another session, is really a clinical trial preparation and really trying to make sure that we have an efficient clinical trial design, which is really crucial to attract companies into working in CMT. So I just want to give you a quick overview of what CMT1 really consists of. When we talk about the nerves that go from spinal cord down to all the muscles, including your big toe, these are pictures here at the bottom that indicate really these axons, which carry action potentials or nerve impulses down to your muscles. And surrounding those axons is myelin, which we sometimes refer to as insulation, and hence the diagram on the far right showing wires. But myelin is more than insulation. It actually makes these nerve impulses go a lot faster. One of the things I like to say is that myelin makes nerve communication 3G into 4G, or if some of you have a 5G phone, it's like going from 4G to 5G. And so myelin is really important. It's also important to stabilize those axons. These are really the longest cells in your body. It's a single cell that goes from your spinal cord all the way down to your big toe. And these cells, because they're so long, are vulnerable. And over the lifetime, you really want to preserve these as much as possible. So myelin plays a really important role with that. Typically, when you talk about CMT1, this is affecting the Schwann cells that make that myelin. And CMT2 is really more of an issue affecting the motor neurons or sometimes sensory neurons more directly as well. But loss of axons and that communication is really a fundamental part of all types of CMTs, so there are some commonalities. So the major types of CMT1 include CMT1A, which is the biggest class. This is caused by gene duplication of PMP22. We have excess of this gene and excess of the protein made by that gene. There's CMT1X, also quite a large percentage of individuals with CMT caused by mutations in the GJB1 gene. This tends to affect males more severely than females. And then CMT1B, also a large segment of CMT, is caused by mutations in the MPC gene. And within all of these categories, and particularly 1B, there's really a variety of presentations. Some individuals are more affected more severely than others that are more mildly affected. So we've been hard at work trying to identify new therapies or ways of identifying new candidate treatments for CMT1. One of those is the drug testing with pharma partners. So we've set up animal models that we make available to companies so that they can test candidate drugs in animal models of CMT. And also some companies come to us, so they've already done their testing and they just want support in other ways, and you'll see a couple of examples of that within this session. There are ongoing studies in the world of CMT involving new classes of drugs called HDAC6 inhibitors. There were a few presentations this summer at a major conference in our field called the Peripheral Nerve Society, and showed some really promising results in models of CMT1A. In our lab, we've developed targets that we can use to reduce PMP22 expression, and we are in the process of testing that based on some new project that's just starting in my lab as well. There's a lot of work to try to figure out if we can make that excess protein go away. And this has been work that was initiated in Laura Feltry's lab in Buffalo, and has been carried on by one of the students in that lab, who's now set up his own lab as well here in the DC area. And this is a promising treatment that could work for both 1B and 1A based on the preliminary results. And then another really promising area is UPR pathways that look like they have some promise in CMT1A and 1B. And in fact, stress pathways in general is an area that seems to be very important for CMT. And in fact, Rob Burgess has a work in another kind of stress pathway that he'll share a little bit about as well. So one of the things that's really been an exciting development in the neuromuscular field is new therapies for spinal muscular atrophy. And this has really laid down a template for us in order to try to develop similar therapies for CMT. Spinal muscular atrophy affects the same motor neurons that are affected in CMT, but this is, tends to be a very devastating neurological disorder that really affects children and adolescents very profoundly. And what's been very encouraging, there are now approved treatments for SMA. Two different companies. One of those uses AAV gene therapy model, and another one is an ASO-based therapy, and there's other companies that have developed other versions of these sorts of treatments as well. And this has really been showing really dramatic effects, particularly in certain types of SMA. And there's also have been ongoing clinical trials for GAN. This is a type of CMT that's fairly rare, but this has been a very encouraging area as well. And so we've been involved in applying these sorts of technologies and trying to develop these to the point where we'll have clinical trials in CMT and 1X. And Dr. Kleopis is going to show you a little bit of his work. He's been a real leader in this area. I did want to highlight one area, which is really this ASO or RNAi. This is a really powerful technological development where we can identify the problem gene and really try to target that specifically rather than affecting enzymes. And one of those technologies is called ASOs, or antisense oligonucleotides. And we were able to work with a company known as Ionis. And using a couple of different models of CMT1A, we really got a dramatic reversal of the symptoms. And we're careful to frame expectations here, but it not only sort of stabilized the neuropathy in these animal models, but it actually allowed for some improvement as well. And whether or not that happens in human clinical trials is certainly an open question. We won't know until it's tried. But this has really been an exciting development, and I'll say a little bit more about that in a subsequent slide. So overall, we have a lot of sort of genetic therapies that are sort of in process. Many of you have heard about CRISPR-Cas9, this really revolutionary technology that's inspired a lot of hope for a variety of different diseases. We collaborate with a company known as Toolgen. They've been doing preclinical testing with us. We've been working with companies on ASO and RNAi. And then also there's a gene therapy version of this using AAV9. And Dr. Kleopis in his recording will tell you more about his work. And this is together with Scott Harper at Nationwide Children's. And this is really, they're driving towards a clinical trial for this in collaboration with Armatus, which is a company that's sort of taken that on. The other thing that is very key for these sorts of technologies is that we have to get them to the right cell at the right time. And so delivery technology is really a major issue. Even if you can get this to work in cells in a dish, that doesn't mean you can deliver it to the right cells in the body. And so we've also funded projects related to delivery projects to get these genetic modification methods into Schwann cells so that they can actually do that work. Finally, I just want to wrap up a little bit just talking about biomarkers in clinical trial preparation. This is going to be discussed in a session by Dr. Shai and Dr. Herman a little bit later. But one of the key things that we've realized is that even if we had the very perfect cure for a given type of CMT, it may never make it to clinical trials unless we have really a good clinical trial design. And so we realized that we really needed to invest in trying to develop ways to make clinical trials more efficient, be able to use smaller numbers of patients, and ultimately cost less money, because if we have a huge amount of financial outlay that has to be done for every clinical trial, that's going to be a huge barrier to bring these things into trials. And so we've been working very hard to try to identify what I like to say as treatment responsive biomarkers that would give companies information about whether a given treatment is actually working, ideally in an early phase of a clinical trial. And Dr. Shai and Dr. Herman will say more about this. We've had major funding from the CMTA to do this in CMT1B and 1X. There's been general funding from the CMTA to the Inherited Neuropathy Consortium, which has also brought forth more studies in CMT1A, and we're now just putting together a larger package of CMT1A studies for this. One of the exciting developments is actually using MRI of calf muscle. So one of the things that seems to be a very sensitive measure of progression in CMT is how calf muscle changes over the lifetime. And using very sophisticated MRI techniques, there's a way to really measure this precisely. And there's a certain amount of change where the muscle eventually changes into other types of tissue. And if you measure that change, that is something that you could see to see if that change is being altered in response to a treatment that's successful. So that's one example of that. I finally just wanted to say that, we like to think of therapy development as a sort of a straight line, that we make really great plans and everything works the way that we think that's going to work. But it's always a little bit more of a windy, curvy road here. And I just want to take one example from the history of things that we've been involved with, just to illustrate that. So I told you that there was FDA-approved therapies for SMA using ASOs. And we worked with Ionis, this company that uses ASOs, and to get preclinical models of CMT1A, and they really showed great success, and this was published. But this really gave rise to a major challenge. Really, one is to have a better, efficient delivery of ASOs to nerve and to Schwann cells. And the other challenge at that time is we really didn't have great clinical trial measures in order to power a clinical trial design. And so these two challenges really gave rise to a number of different things. First of all, the success of Ionis has inspired a whole series of companies, some of which we can name and others that are yet unnamed because they want to keep their efforts confidential for now. But one of the leaders in this has been a company known as DTX Pharma, which has been bought out now by Novartis. And we're hopeful that that will bring this into clinical trials, hopefully in the next 12 to 18 months or so. The other thing that this inspired is we realized that we didn't have adequate clinical assessments to really power a clinical trial. And so this really inspired us to bring on some new kinds of studies and other things that we can incorporate into a clinical trial to make that more efficient. And the combination of those challenges and the responses to those challenges, I think, has now put us in a position where we're really able to kind of see how we can have a well-designed clinical trial. And we're very hopeful, based on the success that was shown by Ionis and the other companies that are working in this area, that this will really create an effective therapy of CMT1A, But of course, we're not putting all of our eggs in that one basket. We're working with other companies that we'll hear about more in this session, and working at several fronts to try to bring more effective therapies to CMT 1A and 1B and 1X as well. So we're working on all of those things sort of in parallel. So with that, I'm going to start to turn this over to my colleagues here. I think the very first one is by Dr. Cleopa, who's a neurologist in Cyprus, who's been really leading the charge in gene therapies for CMT. And he didn't come over from Cyprus, but he was gracious enough to record a presentation for us, which I understand will be played now.
Good afternoon. My name is Cleopas Cleopa. I would like to welcome you to this session. And I would like to give you a brief update on our work to develop gene therapy for CMT1A and CMT1X. CMT1A, as you all know, is the most common cause of CMT neuropathy and accounts for about 60% of all cases. The disease results from peripheral myelin protein 22 gene duplication. The presentation is typically with weakness and wasting of distal muscles, difficulty with walking, loss of sensation, impairment of balance, and the disease is very slowly progressive over the lifespan. PMP22 is an important protein in the peripheral nerves, expressed by the Schwann cells, which form the myelin sheath, and it plays an essential role in the regulation of myelin formation, but also maintenance throughout life. In CMT1A, we have a duplication of the gene resulting in 3 instead of 2 copies of the gene, and that leads to excessive overproduction of the PMP22 protein, leading to accumulation within the cell with further toxic effects, dysregulation of the myelin integrity, and to demyelination. So based on this gene dosage effect, all therapy approaches that have been designed to treat CMT1A mainly target the PMP22 gene overexpression and try to reduce the excessive amounts of PMP22 protein. We have several non-viral methods that use liposomes, nanoparticles, and antisense oligonucleotides. One of the most promising technologies is the fatty acid ligand conjugated oligonucleotide, the Falcon siRNA, which has been developed by DTX Pharma and currently further developed by Novartis, hopefully moving to clinical testing in the next year. We also have nanoparticle-based approaches with squalene and other formulations. We have several viral vector approaches, including our program. We use adeno-associated viral vectors that are able to deliver silencing molecules, microRNAs, short hairpin RNAs, but also CRISPR editing technologies to modify the PMP22 gene. All of these therapeutics can be delivered by different methods, including subcutaneous injections, mostly used for ASOs and nanoparticles. We use intrathecal delivery for the viral vectors, and there is also intramuscular delivery, mainly used for trophic factors. Our program to develop a gene silencing approach for CMT1A has been based on the development of a microRNA that specifically targets the PMP22 gene product. This has been designed in collaboration with Scott Harper at Nationwide Children's Hospital, and we packaged this nucleotide into the adeno-associated viral vector 9 and delivered it by lumbar intrathecal injection into the CMT1A model that overexpresses the human PMP22. We demonstrated widespread expression in the peripheral nerves in this model. And we also showed that this can lead to specific reduction in the gene product levels of the PMP22, the mRNA levels, whereas other myelin-related genes are not affected by this nucleotide. We also showed the same effect at the level of the protein, where we had reduced levels of the PMP22 protein compared to the untreated animals. When we tested this therapeutic silencing RNA in the model of the disease, we showed that both when delivered at the young age, at the early stages, but also at the older stages of the disease, we can achieve improvement in motor function measured by the grip strength and the mobility duration. We also showed improvement in the motor nerve conduction velocities and also in the amplitude of the nerve response in the peripheral nerves. When we looked at the morphology of the peripheral nervous system, we also found improvement in myelin sheath formation, shown here, in contrast to the untreated animals, which have many nerve fibers that are completely devoid of a myelin sheath. This program is currently moving towards translation with support from Armadus Bio. We developed a second-generation clinical stage vector that can be used for human treatment. We also performed a dose escalation and efficacy and safety study, examining different doses of the vector. And this work showed that we have a dose-dependent transduction of the cells of interest, a dose-dependent effect on the PMP22 silencing, and also a dose-related therapeutic benefit that was best when we use the same dose used in our previous work. So this therapeutic silencing vector is now tested in non-human primates. This is a major step forward, and it will show the potential to scale up the treatment in humans and also, importantly, to demonstrate safety. And hopefully, if we get positive results from this study, this can lead to further FDA application planned for early next year. In addition, in a project funded by CMTA, we try to optimize further the gene silencing approach for CMT1A. We want to identify novel vector capsids that may be more efficient than AB9. We tested several of these capsids in collaboration with Dr. Steve Gray, and we identified two of them that appear most promising for targeting Schwann cells. We also tested various DNA elements to use cell-specific promoters that will make the silencing specific to Schwann cells in collaboration with Dr. John Svaren. And after testing several of these promoters, we identified the MPZ promoter as the most reliable to control the expression of the silencing RNA. So now we want to combine these with the new capsids and do a final testing in the CMT1A model.
Hi, I'm up next. I'm Robert Burgess, and I'm at the Jackson Laboratory up in Bar Harbor, Maine. JAX is a nonprofit research institution, and we specialize in mouse genetics and making animal models of human diseases. And hopefully we got a couple of slides here. And if not, it doesn't really matter because I can just tell you everything. But we're working on a variety of approaches using these animal models, and we're doing gene therapy approaches of the sort that Dr. Cleopa just described. But those approaches, almost by definition, are going to be specific to a given subtype of CMT, right? Reducing PMP22 is going to be effective for CMT1A, or putting back a gene will be, SORD1, for example, would be effective for that. But those gene therapy approaches are unlikely to be beneficial across multiple CMT subtypes. So one of the things my lab has been interested in, and a couple of these projects have been funded by the CMTA, is trying to find more generalizable approaches that might work in a variety of different subtypes of CMT. And one of those approaches that John mentioned earlier is the use of HDAC6 inhibitors. The rationale for this is that, as John mentioned, the cells of your peripheral nervous system live in the spinal cord, but they have to send out these long axons all the way to your hands and feet. So there's a fair amount of traffic actually back and forth between both the periphery back to the cell body and from the cell body out to the periphery. And HDAC6 inhibitors should enhance that trafficking back and forth. And it's one of the things that at a cellular level goes wrong in CMT. So there's a really strong rationale for doing that. And there have been really promising preclinical studies in a variety of mouse models that using HDAC6 inhibitors can be beneficial in probably 4 or 5 different types of CMT now using animal models. So an interest in further expanding that, finding out how many different types of CMT might actually benefit from an HDAC6 inhibitor, and then eventually moving that into trials. Another strategy that my lab has been working on is inhibiting a protein called SARM1. So SARM1 is really interesting and somewhat counterintuitively, it's a protein that actually promotes the degeneration of peripheral axons. This is probably important for regeneration after injury and under normal circumstances. But it's also not unreasonable to think that inhibiting a protein that usually promotes axon degeneration could be beneficial in diseases like CMT. And inhibiting SARM1 has actually been shown to be really beneficial in things like diabetic neuropathy or chemotherapy-induced peripheral neuropathy. And we're now exploring inhibition of SARM1 in a variety of the CMT mouse models that my lab works with. So we're hoping that we can find at least a subset of forms of CMT that might benefit from SARM1 inhibitors. And there's a big push in the pharmaceutical industry to develop these SARM1 inhibitors. So hopefully they can identify the drugs and we can identify the target diseases. Oh, there we go. All right. So that's what I'm telling you. And then the third area that my lab is really actively involved in is targeting cellular stress pathways as a strategy for CMT. And John mentioned this. This is certainly relevant to CMT1B, for example, and the work that Laura Feltri and Maurizio D'Antonio are doing. But we've also found in some of the subtypes of CMT that we work on, cell stress pathways are activated. And it seems that inhibiting those pathways actually can be really beneficial in some sorts of CMT. But it's also going to be a little complicated because in the form that my lab works on the most, which is actually an axonal form of CMT 2D, it seems like we need to keep those stress pathways off. And in things like CMT1B and the work from Italy, it seems like you actually want to keep those stress pathways on. So it's not going to be still not going to be a one-size-fits-all therapeutic strategy. But the good news is there's, again, lots of efforts in industry to develop drugs that will keep these stress pathways either on or off for a variety of different conditions ranging from cancer to Lou Gehrig's disease. So there are candidate drugs that are already being developed, and my lab is actively involved in trying to test those in some of our CMT models. So we think that that's actually going to be a beneficial strategy. Since I'm at the Jackson Laboratory and really specialize in mouse models of CMT, I added it up the other day. I think I have 14 different forms of CMT with mouse models. And I'll be totally honest, I think 7 or 8 of those are really, really good models that look a lot like the human disease. And 3 or 4 are okay and recapitulate certain parts of the human disease, and then 3 or 4 of them really stink. They don't look anything at all like the human disease or don't do anything at all. So it's not a slam dunk that if we just engineer the mouse, it will look like the patient's, but it does work most of the time. And there are a variety of reasons that we want to do that. First of all, we can really go in depth in our analysis of the mice and identify potential therapeutic strategies. And these cell stress pathways are an example of that. We didn't know that those pathways were being activated until we studied the mice. And then we also didn't know if those pathways were good or bad, right? Is this pathway turning on a good thing that's saving the peripheral neurons, or is it actually a bad thing? But we were able to address that using animal studies to determine which way we wanted to tweak this pathway. John mentioned that FDA wants to see this preclinical data, as it's called, in animal models before they will move to an investigative new drug application. So it's actually really important data for us to be generating, and the better the model, presumably the better the data is going to be. And fortunately, we have lots of good animal models of CMT. There are very good animal models of 1A, 1B, 1X. The big ones probably for people in this room, but actually many others. So not all of them, and sometimes rat models turn out to be better than mouse models, and I wish I knew why. But we're not usually too limited by lack of animal models. Sometimes nobody's tried to make one yet. There are 100 genes involved in CMT, but for the most part, we actually have good animal models for most of these, and that should really speed up the preclinical research and the ease of getting something into a clinical trial. So we've seen lots of patient movies and gait analysis, and sometimes these animal models maybe are a little obtuse, but I want to show you a quick movie of my patients. So if we can play that, I'm not sure if I can do that. So this is actually the first mouse model that I found. Hopefully it's going to play. What's that? Oh, no. All right. So you were about to see a mouse that was the first mouse that I found that had Charcot-Marie-Tooth disease. Yeah, it's just not going to go. Classic problem. But the mouse has all of the hallmarks of the human disease. It has gait defects. Instead of running around on all fours, he's kind of belly crawling and paddling around. That reflects weakness and poor muscle tone in the limbs. His tail is kind of flopping behind him like a wet noodle instead of the way a mouse usually runs around, again, reflecting poor muscle tone. It's a length-dependent axonal neuropathy. It progresses with age. It has really all of the salient features of human Charcot-Marie-Tooth disease, and it's really proven to be an excellent model for us. Oh, here we go. All right, good. Okay, so the guy on the upper left, that's the normal mouse on the left, and then this is the CMT mouse. So I hope I can convince you that that's what CMT looks like in an animal model, and these animal models can actually really recapitulate the human disease very well. And this one in particular is actually quite an early onset disease. So that mouse is only 3.5 weeks old. That's an adolescent mouse. So it actually also makes our research very efficient. I can run studies in 6 weeks or 8 weeks on these mice and know if I've had a good effect or not, because the onset of that phenotype is so early. We could watch the mouse for another minute, but this is another mouse with the same thing. But anyway, I'd like to thank you very much for inviting me down here and for your attention. And after the presentations, I'd be happy to answer any other questions. Thanks.
So Dr. Cleopa is a force of nature. He's not only been working in CMT1A, but I think we have a video of his work in CMT1X.
1X is the second most common type of CMT neuropathies and results from mutations in the GJB1 gene that encodes for a gap junction protein known as connexin 32. This is an X-linked disease, and therefore men are affected earlier and more severely with a typical phenotype similar to that of CMT1A and slow progression. Connexin 32 forms very important channels of communication that traverse the myelin sheath in the peripheral nerves, and there it serves the homeostasis and the support of the axon, and in end effect, the integrity of the whole peripheral nerve. mutations in CMT1X have been shown to disrupt the function of connexin 32 and cause loss of the gap junction formation. So based on this knowledge of CMT1X pathogenesis, there are different treatment approaches. Our approach is to use gene therapy to replace the healthy connexin 32 gene using a cell-specific promoter element. There are also non-specific neurotrophic factor-based approaches delivered intramuscularly, and also some drug-based therapies that either target inflammatory cells or aim to prevent axonal degeneration that occurs as an end effect in this disease. In our program, we use an adeno-associated viral vector-mediated connexin 32 gene replacement. We use a myelin-specific promoter to drive the expression of the connexin 32 specifically in the myelinating cells, and we deliver the vector by lumbar intrathecal injection. Using this method, we demonstrated that we can achieve expression of connexin 32 in Schwann cells throughout the peripheral nervous system, and we performed treatment trials in different models of CMT1X, including the connexin 32 deficient mouse, but also 2 transgenic animals that express CMT1X mutations. We showed with this treatment, both at early as well as at late stages of the disease, that we can achieve improvement in muscle strength, in nerve conduction velocities, in myelination of the peripheral nerves, and also we can reduce the inflammatory changes. In the last 2 years, with support from the industry, we also optimized the gene therapy vector for CMT1X. We used the AAVrh10 vector, and we tested different doses in a dose escalation study, showing that we get a dose-dependent therapeutic benefit, shown here with the muscle force, but also in the nerve conduction velocities. And this program is also moving now towards further testing in non-human primates. We also tried to develop alternative non-viral based gene delivery approaches because we want safe and efficient access to the Schwann cells to deliver the therapeutic gene. So in a project that has just been funded by CMT Association and Muscular Dystrophy Association, we develop a hybrid nanoparticle that will encapsulate the therapeutic gene under the MPZ promoter. We will conjugate to these nanoparticle recognition molecules that will facilitate the entry into the Schwann cells, and then we will test these nanoparticles in the model of CMT1X. So in summary, we have made some progress in the long pathway towards translating treatments for CMT1A and CMT1X. The starting point is the good understanding of the disease cause and mechanisms for these diseases. Based on this understanding, we developed the therapeutic approaches that were tested in proof of concept studies showing promise in the models of the disease, both for 1X and 1A. We further did dose escalation and toxicity studies in both programs, confirming the proof of concept studies. And we also tried to optimize the technology by using alternative capsids, more cell-specific technologies, and also non-viral methods. Currently, we're making a big step forward by testing in non-human primates. For both programs, for the CMT1A, testing has been ongoing since the summer, and for CMT1X, has been initiated in October. This is very crucial for demonstrating the potential to scale up treatments to humans and also their safety. And hopefully, if this turn out to be positive, will lead to clinical trials for which, of course, we need to be ready. We have to have natural history studies, sensitive biomarkers, and this is what you will hear about more in the subsequent sessions. And this will also facilitate successful approval of therapeutics for treating patients. I would like to thank you for your attention and also acknowledge the members of my team for doing all this work, also the contribution of many collaborators in these projects, and of course the funding by the CMT Association and other organizations that made this work possible. Thank you again.
I will convey your appreciation to Dr. Cleopa. He's really an amazing scientist and clinician. So we're very grateful to have Dr. Zhukovsky here from Agoronics, and we'll move to his talk next. And thank you so much for coming.
Great. Good afternoon, everyone. Thank you very much for the invitation to speak today. It's a real pleasure to be here with you in person. So today I want to introduce you to a molecule called alpha-1 antitrypsin. And that's a molecule that we believe can really help CMT1A patients. And just to give you a brief introduction of what this molecule is, it's what they call alpha-1 antitrypsin, which is a biological compound. And we all have it. So it's present in normal physiological concentrations in our body. So that means it is very, very safe. It has indeed been approved in 1987 for being therapeutically applied in a disease called AT deficiency. So some patients don't have enough AT in their body, and it is a replacement therapy that is available for those patients. And the currently prescribed dosage for those patients is 60 milligram per kilogram per week. But we believe in our studies, and we found that doubling this dose to 120 milligram per kilogram per week can really help improve the symptoms for CMT1A. And that's based on studies that we've done, preclinical studies that we've done on mice. That's achievable lower biomarkers, including pro-inflammatory cytokines. Let me just explain to you our strategy and how we arrived to understand that AAT might be helpful in CMT1A. As we all heard, PMP22 is very, very important in regulating or bringing about the disease of CMT1A patients. Now, there is a study from Johns Hopkins University that really tried to understand, so we know PMP22 is bad, but what does it actually do to the cell? So everybody is now working on gene therapies and saying, we need to lower PMP22 and then we have a cure. But we tried to build on something else. So we tried to do kind of a middle solution. What does PMP22 really do? What does it cause? And what happens to the cell? And the answer came from this, the partial answer came from this research from Johns Hopkins and saying there is real increase in inflammation, so pro-inflammatory cytokines. So your cell really doesn't like it when there's too much PMP22. But can we trick the cell to thinking that the PMP22 is no longer there, or at least there is no inflammation? And I just want to give this analogy here of what I believe is PMP22 does. So you can think about it as the hot desert sun, which doesn't allow for anything to grow. Now, if you really want to, maybe gene therapy is more like changing the weather. So you can bring clouds, and the technology allows for it. What we're proposing is doing something more local. So watering the tree in the desert, for example, could be one example of how AT works to really get the myelin growing and to protect the myelin sheath. So really applying this local anti-inflammatory, water in this case, and then for an apple tree, for example, to allow it to grow. Just want to expand on that a little bit on the kind of the biological level. So if you think about PMP22 overexpression, you have different cytokines that get upregulated, such as interferon gamma, interleukin-6, interleukin-8. And AAT is able to regulate all of those and downregulate in the Schwann cell. But what is important to understand is Schwann cells is not the only cell involved in the disease. There's also macrophages that are very, very important, and they are not only responsible for propagating the inflammatory response and attracting more cells that damage the myelin and damage the axons, but in fact, if switched, they can actually help to restore the axon. So here we showed in our experiments in vitro, in the lab, that AAT is not only able to reduce inflammation in Schwann cells, but is also able to switch macrophages from their M1 state to M2 state. So going from bad macrophages to good macrophages and restoring axons. And just, in the interest of time, I'm going to give you some of the headline results. And this is maybe a bit oversimplified, but we measured the compound muscle action potential, which is really the health of the axons, the thickness of the myelin sheath, which is also represented by the nerve conduction velocities, and the grip strength. And this study was done very shortly in mice, only 2 weeks, is because this is a human therapy and this is a human disease, but we all try to model it in a mouse model, and we can see that the mouse model really represents the disease parameters. But here, within those 2 weeks, we can improve it almost halfway to healthy. So improvement. So if you have a normal CMAP of 8 and the diseased one has around 4, we managed to get it to 6 in only a matter of 2 weeks. So now, how does it translate into humans that needs to be studied in clinical trials? But the results in mice were really compelling, and I'm really excited to share this data with you today. And then the question is, so what's next? So it's, we all love the story, it's all fantastic, and what happens next? So we did share our results with the FDA, and we received the orphan drug designation this summer in July, so very recently. And of course, the next stage for us is to apply to the FDA to get the drug approved for use for CMT specifically. So yes, as I mentioned before, 60 milligram per kilogram works for another disease, but we believe that we need to double the dosage to be effective in CMT. And this starts a very long process until this is available to patients, and hopefully all goes well. But the good news is in between, we are looking to recruit for a clinical trial. Hopefully, if everything goes well, that will be in the end of next year. So please do visit our website, which you can see here. You can register your details and get the updates of when we're launching. We're thinking of doing this between Switzerland and the US. So I'm from Switzerland. I'm based in the University of Geneva Hospital, which is the largest hospital in Switzerland. And I hope that many of you can be enrolled in our clinical trials. Just want to take this opportunity to thank my team. So we are tirelessly continuing to work to find ways to improve it and ways to measure and work with the CMT Association to really make sure that the clinical trial works and that we design it in a way that we can get medicine to you as soon as possible. So thank you very much for your attention. I really look forward to your questions. Thank you.